Hello, my name is Maithreyan Srinivasan. I'm the CSO for Scribe Biosciences. The title of my talk today is Microenvironment on Demand, a breakthrough technology for personalized cell therapy discoveries. Scribe Bio was spun out of the Abate Gartner Labs at UCSF, and the technology that we use is a proprietary droplet microfluidic platform for the precise construction of single-cell co-culture assays for high-throughput cell-cell interaction analysis and linking those functional interactors to single-cell sequencing. We've had 3 publications so far, and our core technology has been awarded a US patent. For the talk outline, I have 2 broad segments. The first part of the talk, I'll focus on the importance of cell-cell interactions, specifically at a bi-cell level. What I mean by bi-cell is typically a sender cell and a receiver cell present in the same reaction vessel, or controlled cell numbers, meaning having 5 cells of one type and 1 cell of another type, though I would not focus too much on that in this talk. And there are benefits to doing the single-cell functional analysis methods, because you could link them, to targeted sequencing of immune cells that matter, and how that's done is what I will talk about in this presentation. And there are many applications that one could utilize this technology for: screening for tumor neoantigens and cognate T cells, gamma delta T cells, gamma delta T cell CARs, screening for those kinds of molecules, and then finally CAR T cell variant screens, which is what I would focus my talk on. This is a general purpose technology, and anytime you want to study a sender cell and a receiver cell interacting with one another, and then link them to single cell genomics, this will be an ideal technology to do that. And then the second part of the technology, second part of the talk, I'll focus on Scribe Bio's microenvironment on-demand technology. I'll talk about the workflow and then the data that we've obtained using healthy donor CAR T cells. So CAR Ts are hybrid molecules that are formed from the variable regions of antibodies, which are linked to form a single chain and fused to T-cell co-stimulated domains and activation domains. And CAR T-cell therapy has been shown to be effective for certain blood cancers. And there has been a lot of interest in deploying CAR T-cell therapy for solid tumors as well. And the main reason for that is they are HLA independent, and so do not require the need for antigen presentation. However, side effects exist for CAR Ts, including cytokine release syndrome and neurotoxicity events. And so the field has been working on how to obtain an optimal CAR. And so the need for CAR T functional screens starts to become a reality when you want to deploy this kind of a technology, this kind of a therapy to multiple cancers. And there is no good way to do this today. So I present a use case here where if you want to screen 200,000 scFv variants and find what an optimal CAR variant, how do you go about doing it, and what is optimal? For example, do you want to check for tonic signaling? You want to look for cell killing efficiency. You may be interested in understanding gene expression differences between optimal and suboptimal CARs and how they contribute to side effects. And these kinds of projects are already ongoing, where I show the evidence in the form of a paper published in 2013, where 150,000 scFv variants were at 100x redundancy were screened. And so this means that you have to screen over 15 million cells, which means that it requires a tremendous amount of throughput. And doing this in a bulk assay, you would have to do it in a serial fashion. In addition to scFv variants, there is tremendous interest in understanding other properties of CAR Ts, such as CAR T persistence in cancer patients, and to understand and improve the persistence of CAR Ts. Co-stimulated domains have been swapped into the CARs to figure out which CARs persist in patients for a longer time. And obviously, one could imagine combining the 2 efforts to screen for optimal CAR variants that have multiple properties. And the way it's done, as I said earlier, is through bulk assays, where thousands of CAR variants are interrogated at a single time, and then in a serial fashion, you understand which variant it is. But this is much easier done using single cell functional assays, provided you have the throughput in the hundreds of thousands to millions, and also have the ability to integrate the functional assay to sequencing, where you can readily obtain the variant sequence information that truly matter. So at a single cell level, people would want to measure cytokine responses, track T cell activation signatures, or assess cell death. And these kinds of activities can be performed using FACS as well, because you can sort a sender cell and a receiver cell into the same reaction vessel. However, FACS-based technologies sort into 96-well or 384-well plates, which utilize larger volumes, meaning microliter-sized volumes. And single-cell secretions are at a very low concentration. As a result, you do not have the sensitivity to assess single-cell assays. And then, as I referred to earlier, once you know which variant functionally is critical, you can take that and you should be able to sequence that. And so you start, this kind of a streamlined technology workflow can help you start from a large number of variants and end up with hundreds to thousands of variants that you can further study for optimal properties. So typically, these kinds of studies, if you want the kind of throughput that we're talking about, are done using droplets or nanowells. And so when you want to do this with droplets, you start with patient T cells, target cells, and immunoassay beads, and randomly load them onto droplets. And when you do a random loading, it's governed by Poisson distribution, and it comes with as many inefficiencies. And so the assembly that you're really interested in, which is a single T cell, patient T cell, and a target cell, and an immunoassay bead, happens very infrequently. And this is shown in a publication where using randomly loaded wells, where some wells get a single cell and other wells get 6 or 8 cells. So what you would really want to do is to take the T cell library, target cells, and immunoassay beads, and of course randomly load them onto single cell droplets, but then load them in limiting dilution such that only one droplet out of 10 or 15 contain a cell or a bead. And then if those cells and beads are fluorescently labeled, then you can sort them and then deterministically combine them into a single assay droplet, and then make millions of identical replicates that will enable you to perform these massive screens. And the workflow for MARD that enables you to do that is shown on the slide. So we'll start with an input mixture where you have the individual cells that are fluorescently labeled, encapsulated in droplets, and then you flow these droplets into a microfluidic channel where it's interrogated by laser excitation. So say the intended droplet is detected, the droplet is diverted into the bottom channel and retained in a dielectrophoretic trap shown by the two dark squares linked by a line. Let's say that the first droplet contained an immune cell that got retained by the trap. The system will wait for the target cell or the reagent bead to appear, and if it sees that a target cell or the reagent bead, it will be diverted into the bottom channel and merged in the dielectrophoretic trap and retained. And once the assembly of a single immune cell, a single target cell, and a target bead or assay bead is encapsulated in a droplet, it gets released and it gets collected, and you can collect millions of this in a tube, and then incubate them for immune cell activation. So when an immune cell is activated, it secretes cytokines, and the assay bead contains primary antibodies for specific cytokines that can be detected. And once the immune cell activation is completed, you have two options to perform single cell RNA sequencing. The top option shows you sequencing using third-party devices, and the bottom option would be using the ScribeBio workflow. So let's first talk about the top option, and I'm going to show you data only for the top option today. So in the top, the workflow on the top shows the same kind of sorting mechanism that I talked about earlier, but instead this time we are going to sort based on the assay positive signal that resulted from the immune cell activation. And so you could collect the hits and then feed it into a 10x genomic system or other third-party device. When you do this, you lose the linkage between the sender cell and the receiver cell, and because when you sequence them using the 10x genomic system, they're all sequenced as single cells, but but the link between the two cells that resulted in the immune cell activation is lost. But if you want to preserve the linkage, then we are working on a method where we have droplets that contain DNA barcoded beads. So each droplet will contain a single DNA barcoded bead, along with lysis and RT reagents that can be flowed through the microfluidic channel, and then using the same mechanism that I mentioned earlier, can be merged at the dielectrophoretic trap and collected, and then you can perform a reverse transcriptase reaction to generate the cDNA. By doing this method, you can, because each droplet contains a single barcoded bead, the information between the sender cell and the receiver cell is coded or by the same barcode. And so this will be important for understanding, say, new antigen, which new antigen is paired with which T cell receptor. So in addition to performing immune cell activation assays and linking them to single cell genomics using collected droplets, we can also array and image these droplets, and that can be used for temporal studies. So to summarize the unique features of this technology, we can perform precision cell combinations, selective high throughput single cell RNA sequencing that's scalable to 1 million deterministic assemblies. We can assemble cells and reagents as long as they are fluorescently labeled. We can assemble them deterministically. And finally, we can array and perform temporal studies. So the next couple of slides, I'm going to show you data to support the individual parts of the workflow. The first part of the workflow is droplet merging. And so for that, we asked the question, if we had a droplet that contained a red bead and a droplet that contained a blue bead, how accurately can we merge them and then get the droplets to have a red and a blue bead in the same droplet? And the results of that experiment is shown here, where we have 104 droplets that we imaged under a microscope, and we manually counted to see how many of those droplets contain a single red bead and a single blue bead. And we found that 93 out of 104 droplets contain a single red bead and a single blue bead. If you observe, there are a couple of scenarios where you have three beads in the same droplet, coming from a two-bead droplet from the initial state, the initial starting point. So those are essentially, they are not artifacts of the merging technology, but rather in the starting emulsion, we had some droplets that contain more than one bead, or two beads, or two red beads, or two blue beads that got merged. And so that resulted in a three-bead droplet. So if you consider that as well as not an artifact of droplet merging, then our droplet merging accuracy is greater than 95%, and the two negatives are shown highlighted by the red rectangles. And the image shows the assembled assay droplets, where the droplets contain a red bead and a blue bead, as we discussed earlier. And finally, I'm showing cells that were labeled with 3 different fluorophores deterministically brought together in droplets. We can see that it's at a very high accuracy that we can bring together three cell types in the same droplet. So the second part of the workflow is direct wash-free ELISA. And for that, what we do is to have a tumor cell and an immune cell, as well as a bead-bound primary antibody for cytokines, as well as dye-labeled secondary antibody encapsulated in the assembled droplet. And if immune cell talks to a target cell, it will secrete certain cytokines, and those cytokines can be captured onto the bead-bound primary antibody, which would enable the dye-labeled secondary antibody dispersed through the entire droplet to coalesce onto the bead producing a focused area of fluorescence. So I want to point out that the total amount of fluorescence in these droplets are the same. Whatever that's present in the dispersed solution gets coalesced onto the bead. And so this slide shows some evidence for the wash-free ELISA. So what we've done here is to encapsulate different amounts of IL-2 protein in the droplets and then perform the wash-free ELISA method that I described in the previous slide. You can see that at 0.1 nanomolar, you can barely see the bead-bound fluorescent. However, at 1 nanomolar and 5 nanomolar, the bead-bound fluorescence is very obvious. And so we think that the dynamic range for the system is anywhere from 0.5 nanomolar to 5 nanomolar, and that is well within the regime of what single cells secrete for concentrations of cytokines in the volumes that prevail in these droplets. And then in the last, the far right, I show the same assay with 4-ball ester activated PBMCs. So in addition to cytokine secretions, we can also measure cell death. And I'm showing an example here where we tried to measure cell death and cytokine secretion in the same assay droplet. So for this experiment, which was done in the early days of technology development, one of the first experiments that we did actually, so as a result, we did not label the CAR-T and Raji cells. We didn't have enough lasers to detect all the different entities that could be fluorescently labeled in this experiment. So we used CAR-T and Raji cells unlabeled. We Poisson encapsulated them. Anti-interferon gamma beads are blue in color, and you can see that in some of the emulsions. If CAR-T cells secrete interferon gamma, the anti-interferon gamma beads that are blue will turn magenta in color. And if you look into at the figure, every droplet that contains a magenta colored bead also contains a green cell indicative of cytokine uptake and suggesting that the cells are dead. In addition to performing cell death and cytokine secretion assays, we can also encapsulate different numbers of target cells with CAR-T cells. So here I show one CAR-T cell paired with either one target cell or 2 target cells or 3 target cells. And you can see that the single CAR-T cell is engaging with up to 3 target cells and forming an nice immune synapse. And so several research questions can be asked using this technology. Ability to study CAR-T exhaustion. For example, if I encapsulate 10 CAR-T cells, 10 Raji cells in the presence of one CAR-T cell, do all of them get killed? So those kinds of questions can be answered. Cooperative cell killing, understanding cooperative cell killing, paracrine interactions, temporal nature of cytolysis versus interferon gamma secretion, tonic signaling. These kinds of functional characteristics can be assessed, and then they can be coupled to single-cell genomics to understand phenotypically how the different cells are different. And that workflow integration is what I'm going to talk about in the latter half of my talk, where I'm going to show you data for how we integrated functional sorting with single-cell RNA-seq and VDJ sequencing using healthy donor CAR-Ts. And so for this experiment, we started with alpha CD19 CAR-T cells, prepared using T cells from 2 healthy donors. CAR-T cells and Raji cells were assembled 1-to-1 and merged with interferon gamma assay reagents in droplets. 20,000 assembled droplets were collected in 1.5 hours and incubated for 12 hours at 37 degrees. And the assay droplets were sorted for interferon gamma signal, and 2,000 droplets were collected. And the droplets were de-emulsified, and single cells were collected, viability measured. The viability was greater than 90% after this process, and subjected to single-cell RNA sequencing and TCR sequencing. For data analysis, we did graph-based clustering, and then using the clusters obtained from the gene expression data, we projected them onto UMAP plots. We computed polyfunctional gene expression signatures, as well as CAR-T, Raji cell signatures, as well as looking at the checkpoint blockade gene expression. I'm showing results from one healthy donor, but the other healthy donor, the results were reproduced. So for expectations, I want to just make sure that what to expect from this data. So hits of sequence of single cells, because for the single cell sequencing, we use the 10x genomic system, and as I mentioned previously, when you put the cells assemblies into the 10x genomic system, they are all going to be sequenced as single cells. The sort population was not washed to prevent loss of precious single cells. And the sort population is also expected to contain some non-hits as well due to suboptimal sorting gate selection. So target cells and CAR T cells are expected to get different cell barcodes, and so they're expected to be in separate clusters. But if target and CAR T cells are doublets, as I showed, like formation of an immune synapse that I showed previously, they will share a barcode between, they're expected to share a barcode between TCR-seq and RNA-seq. So this slide shows the gene expression data that we obtained from our efforts. The left-hand side shows the gene expression, and right-hand side shows the VDJ sequence information overlaid on top of the gene expression data. The UMAP plots were derived from 7 groups of clusters identified by graph-based clustering. Even though you see 7 groups of clusters, what is obvious is there are 2 large subpopulations of cells here in this UMAP plot. So what are those clusters? So we try to drill deeper into that. And so for the first question that we asked was, where are the Raji cell markers? And when you look for the Raji cell markers, they're enriched in one of the subpopulations of the CAR Ts. And when you drill further down into the data, you see that 97% of the cells in these clusters shared barcodes between VDJ and the Raji markers. And so this suggests that potentially these are Raji T cell doublets that happen due to an immune synapse event. Then the next question that we asked was, what about checkpoint blockade genes? And so when you look for checkpoint blockade genes that are highlighted in blue color in the top in the right hand side of the slide panel, you see that they are all enriched for checkpoint blockade genes are enriched in a couple of clusters on the other subpopulation that is not common to the Raji cell markers. And that I thought was interesting. And so when we looked into the data more deeply, we found that 71% of the cells with VDJ barcodes in clusters 4 and 6 express checkpoint blockade genes. And finally, the CAR Ts in clusters 2 and 3 shown here appear to be a separate third group and was unremarkable. So it was really interesting to see 2 subpopulations of CAR T cells, one subpopulation aligning with Raji cell barcodes, and then the other population expressing checkpoint blockade genes and not a whole lot in common with the Raji cell barcodes. So we next we asked the question: what about polyfunctionality? How polyfunctional are these CAR T cells? Because highly polyfunctional T cells within CAR T cell products have been associated with clinical response. So we wanted to understand whether the cytotoxic CAR Ts are also highly polyfunctional. And polyfunctionality is defined by a T cell, any T cell that expresses more than 2 cytokines. And when you look at the cytotoxic CAR Ts that we saw here, they showed low polyfunctionality and not high polyfunctionality. And what was also interesting was the clusters that showed enrichment of checkpoint blockade genes showed high polyfunctionality. So to summarize, I've shown some data that integrates single-cell co-culture assays with single-cell RNA sequencing and VDJ sequencing. Interferon gamma assay-based sorting results in enrichment of polyfunctional T cells. And there appears to be 3 distinct populations of CAR Ts. There is a Raji-associated CAR T that may be cytotoxic and show low polyfunctionality. There are highly polyfunctional CAR Ts that also show expression of checkpoint blockade genes, indicative of T cell exhaustion. Just remember that these CAR Ts were sorted based on interferon gamma sorting. So, why were they sorted based on that signature? It's something that we want to understand better. And then finally, there are CAR T cells in clusters 2 and 3 that are unremarkable. And these results were reproduced using CAR T cells from the other donor. And finally, I want to acknowledge the ScribeBio team for their hard work. They pulled off a lot of data in a very short period of time. I'm extremely thankful for their hard work. And then, of course, the Abate Gardner Lab for the continued support, Chimera Bioengineering for providing us with the CAR T cells from healthy donors, and our seed investors, DARPA, NIH, and NSF for continued support in the form of funding. Thank you very much for listening.